Hello, and thank you for joining us today. We are happy to welcome you to the Human Cell Atlas Biological Networks seminar that is today focusing on reproduction. We would like to remind you to join the HCA. We have 18 biological networks. You can join as many as you want. Please visit our website and make your selection. If you have publications you would like to submit for inclusion as an HCA publication, you can also find all the information on our website. It will help increase visibility to your paper. We welcome you to the upcoming HCA events. The Immune Bio Network will be featured next month on May 12th. We started the registrations for HCA General Meeting, which will be hybrid, held in Vienna, Austria, and also broadcast June 27th through 29th. And the HCA Development and Pediatrics Meeting, also hybrid, will be held in November. Please visit our website and register. The agenda for today's seminar: we have 3 speakers. Each speaker will have 15 minutes and followed by 5 minutes questions and answers. Please put your questions in a question and answer function of this platform, and we will try and answer as the time allows. This will be followed by a panel discussion led by the coordinators of the Reproductive Bionetwork, Sue Hamoud, Roservento Torno, Cecilia Lenskog, and Ariela Shikanov. For those of you who are watching us on YouTube or Bilibili, please be advised that we are only recording the first 2 talks today. The last talk by Dr. Clark and the panel discussion will not be recorded due to the nature of unpublished results that we're planning to discuss. And with this, I would like to ask our coordinators to introduce our speakers. Thank you.
Thank you, Ellen. I'm Cecilia Lenskog, and I will introduce the first speaker, Dr. Pauliina Damdimopoulou, who's a research group leader at Karolinska Institute in Stockholm, Sweden. She has a training in biochemistry, cell biology, and toxicology, and her group investigates the impact of pharmaceuticals and environmental chemicals on fertility in women, with a special focus on ovary. And Pauliina will talk about characterization of human ovaries from birth to menopause. Welcome, Pauliina.
Thank you very much for the kind introduction. And now I don't see the button anymore that I should click to share my screen. Where should I find the share?
Okay,
Pauliina, if you go to the apps along the top,
right,
and then there should be a little pop-up that says start
yes,
screen share. You've got it. Well done.
Thank you. So I hope you have the full slide view now. First of all, thank you so much for inviting me. It's my pleasure to talk about our research here today. As Cecilia said, my name is Pauliina Damdimopoulou, and I am a research group leader at Karolinska Institute. And my research group is called Chemicals and Female Fertility, and we have 4 main aims in our research. We would like to map the exposure of women who live in Sweden to environmental chemicals and then investigate how these exposures affect fertility. We are characterizing on molecular and cellular level how human ovaries look like, and then we would like to check how these chemical exposures affect fertility by possibly affecting the ovaries. And finally, we would like to construct different models for ovaries in vitro so that we could better carry out chemical health risk assessment, maybe develop pharmaceuticals, and perhaps in the future to produce oocytes in vitro. But today we will be focusing on this molecular and cellular characterization of human ovaries. Just as a short background, 1 slide. Ovaries, they make mature competent oocytes and of course also hormones that enable pregnancy. And I start with the picture that shows ovaries from a century ago. This is a cartoon I found at a second-hand market, and I was thrilled to find out it was drawn by Karolina Widerström, who was the first female physician in Sweden. She was specialized in obstetrics and gynecology, and she devoted her life to educating women about their reproductive health and reproductive rights. And that's how she drew the human ovary. If you Google ovaries today, this is likely the cartoon you will find. So you can see that there's not much more detail compared to 100 years ago. If you get the chance to see a human ovary, this is what it looks like. This is almost an entire human ovary freshly from the operation theater. And then if you make a cross-section of this ovary and do an H&E stain, this is what it looks like. And you have to really zoom in close to the surface to find these follicles, primordial follicles that make up the ovarian reserve and define the fertility in women. So now looking back at these cartoons, I'd say that Karolinna at least got the location of the primordial follicles right in her ovary cartoon. So ovaries only function for a limited amount of time, and it is all about these primordial follicles that are formed during fetal development and only decline in numbers after that. And one of our challenges is that couples wait longer and longer before they start building their families. So in the European Union, the average age of a first-time mother is close to 30 years, and there's a wide variation in this number as well. And of course, if you start family quite late, it means that you likely won't be able to have that many children, and the total fertility rates in the European Union are well below the population replacement rate by now. And many of these women might then find out that their fertility is not so good anymore. Some might be classified as infertile, and perhaps also in this situation, the exposure to environmental pollution can have an impact on the fertility too. So I think that in order to understand ovarian senescence and what defines fertility and how we could maybe restore fertility, it is important to characterize what kind of changes ovaries go through during these time periods. So my lab is quite young, and we published our first study on this topic only 2 years ago when we did the single cell sequencing of human ovarian cortex to map what type of cells are present. And I will just briefly explain this paper here. First of all, it's always important to consider what type of tissue has been used, because ovarian tissue is not easily available for research. My group, we can work on cortical biopsies from elective cesarean section patients, and then we get larger amount of tissue from gender reassignment patients, where almost 2 entire ovaries can be donated to research. In this paper from 2 years ago, we cryopreserved all the tissue samples and also carried out careful quality controls to ensure that the histology shows follicles. And for the GRPs, we also carried out follicle viability assays to really know that the follicles are alive despite these androgen treatments that the patients go through. And when we were then ready to do the analysis, we would thaw the tissue and digest it to single cell solution. And here we did the 10x genomic single cell sequencing. We also profiled cell surface antigens by flow cytometry, and we had a special focus on DDX4 antibody positive and negative cells. And we also carried out comparisons to human fetal ovaries. So in brief, we found 6 main cell types in the ovarian cortex, and both C-section and GRP ovaries contributed to all these clusters. So both patient types had the same clusters. Vast majority of the cells were classified as general stromal cells, and the most rare cell type was the oocytes here, and their granulosa cells in blue here. And then there were some immune cells and perivascular cells and endothelial cells in these ovaries. What about these DDX4 positive and negative cells? These studies were inspired by publications from 10 years ago that suggested that adult ovaries contain a rare germline stem cell that can make new follicles. And these germline stem cells could be then sorted out from ovarian cortex using this ABCAM antibody that is targeting the C-terminal domain of DDX4 that in these germline stem cells is on the cell membrane instead of in the cytoplasm as usual. And previously, the group where I worked at as a postdoc had problems in finding these cells, and now we decided to give it a new try with these much better tools like single-cell sequencing. So we isolated the cells. They're typically present in a few percent in ovarian cortex, and then we sorted as many antibody positive and negative cells for the single-cell sequencing. And the results showed indeed that these antibody positive cells, they form their own cluster. They're clearly separate from the other cell types in the ovarian cortex. And when we annotated the identities, it showed that these antibody positive cells are actually perivascular cells instead of germline stem cells. And we did many different things to validate this, and I show you here a staining of human ovarian cortex. You can see an antibody staining. Here you see a blood vessel by CD31 positive endothelial cells surrounded by MGAM and RGS5 positive perivascular cells. And then in the next section, we put the DDX4 antibody, and indeed it labels something in these perivascular cells. And you can see when we zoom out that there's Siemens and follicles in the same section, and these are very positive for DDX4, just like we would expect it to be. And when we do RNA FISH to look at the transcript, the DDX4 probe will label the oocytes as we expect, but not the blood vessels. So this suggests to us that the antibody that is recommended for the isolation of these cells is unspecific. It labels something in the perivascular cells, but they do not express DDX4 RNA, and they do not share any characteristics with germline cells of any kind. Well, that was the story of the adult fertile age ovarian cortex, and now we would like to move on to looking at the full lifespan of ovaries. And while older ovaries are typically quite easily available for research, the younger ovaries are definitely a harder thing to have. And we started a fertility preservation study for girls and young women in Sweden to be able to study child ovaries. The study is called Svea Fertil. It is a multi-center, ethically approved research project that gives the clinical service to all girls and young women who are at high or very high risk of treatment-induced infertility. So all patients between 1 and 17 years of age can join. And we have all 6 university hospitals with pediatric oncology and hematology units in the project. And here, a tissue sample is collected if the family consents and the patient meets all the inclusion criteria. And then 2/3 of the removed tissue will be clinically cryostored for the patient, and that will never be available for research. 1/3 of the tissue will come then for research to my lab, and our purpose is to do a careful characterization of the cell types and follicles to understand if it is similar to adult tissue, because we know already that adult tissue can be collected, cryopreserved, thawed, and transplanted back, and it will give pregnancies. So this fertility preservation by cryopreservation of tissue is an established method for adult women, but not for children yet. So we kicked off Svea Fertil in March 2020. And you know exactly what happened then. That's when the COVID started as well. And no matter what the media has said about the Swedish strategy, not really being very harsh, I can tell you that it did affect research a lot. So we've been now operating for 2 years under the pandemic and only been able to include a few patients. And I show you the very preliminary data here. What we've decided is that we now, first of all, dissociate every tissue directly to single-cell solution and prep it into a library. So instead of cryopreservation, we do it freshly. And for that reason, we are also including new adult samples so that we have a comparable reference. And we, as expected, we have a very wide variation in patient age, diagnosis, and treatments. And as you can see here, for example, oocytes in orange, if you look at the age groups, most oocytes are found in the youngest patients, like we would expect, and also granulosa cells tend to be mostly present in the youngest patients. And then we can see perhaps some differences in the different somatic cell populations, but as I've labeled the data as preliminary, so we need to work a bit more to identify these cell types in more detail. So our ongoing efforts are focused on annotation of the various clusters that we find, and then of course localizing them back into the tissues and trying to understand also the impact of age and treatments and maybe even diagnosis. And also we are now starting the analysis of the perimenopausal women so that we can include the aging ovary into this picture. Another project we are carrying out is a detailed analysis of cortical follicle types. We all know that the 10x single cell sequencing is very shallow. It doesn't give quite that detailed picture of the cells. And we are very much interested in the follicles and how they compare between children and adults. In particular, the primordial follicles that should be the same. They're built in utero and they can last for decades, but are they really the same before and after puberty? I don't think we know. So we have picked 60 follicles from children and 60 from adults from the cortical part of the tissue. We have classified them as primordial, intermediary, primary, or secondary. And here you even see some neutral red stain left. So we are making sure they're viable. We take pictures from each and every one so that we can even go back and see how they looked like. And we've done SmartSeq2 libraries and sequenced them. And now we are looking at how the gene expression changes from primordial to primary and secondary compare between children and adults. Also very preliminary data, but I just wanted to give you a small glimpse. If we use the false discovery rate 0.05 and we look at the transition from primordial to primary, we have a lot of genes that change in expression. And then the overlap in those genes between children and adults, there is some, but it's definitely smaller than the numbers of unique genes. So some conclusions from this short presentation. We have mapped the main cell types in adult human ovarian cortex, and you can look at that data in the reference here, and you can also browse our data by going to eovary.ki.se. I want to emphasize that I always get the question, what about normal ovaries? So I don't really know anymore what people mean with normal ovaries. So first of all, ovaries are never available for research from completely healthy young women. So there's always a reason there's a surgery or parts of ovaries are removed. And anyway, ovaries in any woman and person should be considered normal and representative of that patient. So therefore, we always need to do metadata collections to understand the clinical background of the patient and the donor, and also careful quality controls of the tissue to know what we're looking at, because even within the same patient group, the quality varies a lot. At the moment, we are doing a lot of studies on child ovarian tissue through our fertility preservation projects, Fertile, and our preliminary data suggests that there might be differences in the cortex between children and adults. And I do believe that better understanding of the tissue and composition and changes by age and exposures will lead to better understanding of fertility, how to preserve it, how to improve it, and even build these in vitro mimics of ovaries that could be used for toxicology or even assisted reproduction in the future. And the questions that we always struggle with are, how many donors should we have? How many cells per donor should we analyze to have the full picture? And then when we have something exciting, how do we validate the hypothesis when the research is done on human ovaries? And I want to finish by thanking all the past and present members of the lab. You can see the key people who contributed to these studies underlined, and then all the clinical collaborators that enabled these studies. None of this would be possible if we didn't have the clinicians on our side. So with this, I want to thank you, and I'm very happy to take any questions.
Thank you very much, Pauliina. So everyone, feel free to write questions in the chat. So we have a few minutes per speaker for questions, and then there will also be a longer discussion at the end after all the three speakers. So I just have a first question that relates to what you said about the difficulties to get access to samples. So you have been working on 3 different types of samples, from pregnant women and the transgender, and then also the fertility preservation. So are there any general conclusions you have drawn so far about pros and cons with these different types of samples and possibility to characterize different types of genes?
Yeah, I think it doesn't matter what patient type we have. There's always questions of how representative they are. I think people would consider the C-section patients most normal because they are, of course, fertile pregnant women, but they are 9 months pregnant when we get the sample. So that's the limitation. And they are a bit older, so they seldom have follicles in the tiny piece that we get for research. So the best from the follicle point of view is the young transgender patients, but there the quality varies a lot. And we always need to do a lot of controls to know that there are follicles in the tissue. And also the child tissue, it varies a lot from patient to patient. So yes, there's pros and cons with every patient type.
Yeah, it's good that you have different types of samples at least, so it gives an even broader view.
At least we can cross-refer and validate across the patients, so that's true.
There are 2 questions in the chat so far. The first one is from Ernst Lengel from University of Chicago, asking, what did you learn about stromal cells in the premenopausal ovary?
There is definitely many different stromal subtypes. So in our first paper, we ended up describing the main cell types, but if we increase the resolution in the analysis, we will see different kinds of stromal cells, some that have more extracellular matrix, some that are more proliferative, some that likely might be on their way towards TICA cells. And this is what we hope to look in more detail now that we have expanded the numbers of patients and ages also in our analysis. So I think there's much more to be learned about the stromal cells. That's a very good question.
And then there's another question from Alvaro Monteiro. We notice in your T-SNP plots that there are no ovarian surface epithelial cells. Are they so few that we can't see a cluster, or are they lost during manipulation?
That's also a great question, and one of the critiques we get often. So the surface epithelial cells are so fragile that I think they basically get lost when we cryopreserve the tissue. So that was one of the reasons why we now work with the fresh tissue, and we are even trying to make libraries of these cells separate so that we really know what they express. Because I don't think we have good markers for human ovarian surface epithelium that would clearly identify these cells. So we are looking for them, and we will prepare, as soon as we get the nice sample, we will make like this gentle brushing of the ovary and try to collect the cells for a library. Because really, as soon as you get the ovary, you put it flat on the plastic and you move it around, you've lost these surface epithelial cells. So they are definitely very fragile and they detach easily. But we're looking into that. So very good question. Thank you.
Okay, there are no more questions in the chat currently, but please feel free to post more and we will have a discussion at the end. So thank you very much, Pauliina. And now we will proceed and introduce the next speaker.
Thank you very much. Nice presentation. I am Rosa Arvento, and it's my great pleasure to introduce Dr. Alain Chédotal. So Alain Chédotal is currently a research director at Inserm and a group leader at the Vision Institute in Paris. He received his PhD degree from Pierre and Marie Curie University in Paris and moved to UC Berkeley and completed his postdoctoral research with Corey Goodman, where he worked on axon guidance. He was recruited at Inserm in 1997 and started his own laboratory at the Salpêtrière Hospital in Paris before moving to the Institut de la Vision in 2008. His lab studies axon guidance, neural migration, and angiogenesis, and has pioneered the use of tissue clearing methods and light sheet microscopy to study embryogenesis. He's involved in various HCA-related initiatives as the coordinator of 2 consortia, HUDECA, which is a state-of-the-art interdisciplinary program, including 8 partners, whose goal is to build a definitive atlas of embryonic and fetal human cell types. and Hugo Deca, which brings together leading European academic and industrial experts from 5 different EU countries and aims at describing the cellular composition and organization of the developing human gonads to understand how it changes during sex determination into testes in males and ovaries in females. So I'm very excited to hear a last presentation and see the lovely pictures. So it's all yours, Alain.
Thanks a lot for this. Do you hear me fine? It's okay?
All good, thank you.
Good, thanks. And I won't play the slides myself. Tracy will do it for me because for technical reasons. So Tracy, maybe you can go to full screen. Is it okay? Or... Can you put the full screen? Or... I don't see anything.
One moment. I'm texting with her, Alain. Just 1 second.
Okay.
Okay, Alain, sorry, it was on full screen for me, so I'm not quite sure why it wasn't for everyone else. So we'll have another go. Hang on just 1 second.
Okay, maybe I can start.
Yeah, sorry.
No, I can start with the slides. So as you will see, I mean, my lab primarily works on axon guidance and brain development, but we have developed some technology to also do tissue clearing to make organs transparent or whole embryos transparent. And during the course of this project, we started to work also on human embryos. And what I will present you today is some work we've been conducting the past 2 years or so on the development of on sex differentiation in human embryos. So as you all know, initially the gonads is bipotential. Whether you're XX or XY, it looks the same. The cell composition is also the same, so there's no differences. That's true around all the way up to more or less 5.5 weeks of development. And then there's a whole cascade of events triggered in testes, for instance, by SOX9 expression that will make such that then the gonads will differentiate. And if you come back 2 weeks later, you have clear testes for male, and then you have ovaries for female. So I'm, as Rosa mentioned, we are different partners in a European consortium that is named Hugo Deca. And in this consortium, we try to use modern technologies to try to understand what are the cellular events and molecular events that occur during sex determination in human. So we're getting human samples, human embryo samples from 6 weeks to about 20 weeks. And we're conducting a lot of analysis, so single cell analysis that I won't present today, as well as trying to develop some new imaging technologies to do spatial transcriptomics. So maybe you can put the next slide, please. Yeah, and maybe again the one after. Thanks. Yeah, and here that's just a picture of this consortium. As I mentioned, we come from different countries in Europe, and you have a link here in the bottom left where you can also find information on this consortium. So for today's talk, I will just talk about what we've been doing in my team together with Paolo Giacobini, also in Lille, which is to apply some 3D imaging technique to the understanding of human sex differentiation and determination. So maybe you can put the next slide. There's 4 movies, so you can play the one in the bottom as well. So I just show you briefly, and we can discuss later the technique we're using. We're using solvent to make tissue transparent. So we can here, of course, on the top right, what you have are mouse brains, but it will be the same with any kind of organ. So we take the tissue, we fix it, we bleach it to try to remove the blood, then we dehydrate to remove the water, and we remove the lipids using solvent. So we are left with a tissue that contains mostly fixed proteins. We use then antibodies to do immunostaining, and we do this before the clearing. And once the sample is transparent, the fluorophore are still maintained. And to image in 3D, we use a light sheet microscope. So here you have an example of how it looks in the bottom right. In this microscope, you create a sheet of light, and you will move the sample in the sheet of light, and you have a camera on the top of the microscope that will take an image of every single focal plane. So it's quite nice because you can see on the bottom left, it's a scan that goes through the human embryo that was stained for the vasculature. And you can see that we can really image very large samples. We can image samples that are like a few centimeters. So we can image whole fetuses. And we can also have now a very high resolution. We can image all the way to almost 25x. So basically, we started to collect human embryos and make them transparent. So next slide, please. So here I'm just showing you an old image from Johannes Müller, who described at the beginning of the 19th century the Müller duct. And at the end of the 18th century, Caspar Wolff described the Wolffian duct. And you can see in this drawing, that's how it looks. And if you go to modern textbooks, anatomy textbooks, it doesn't look so different from what Müller and Wolff described a long time ago. So we started to, as I said, to revisit the remodeling of the ducts using our 3D protocol. So the next slide, we show you a movie of the embryo that was stained with PAX2. And PAX2 is a nice marker for all the different ducts. And then we use a software, Imaris, to pseudo-color the different components. So if there's a single antibody, but you can use in 3D, we can then paint the Müller duct here in magenta, the Wolffian duct in blue, and the kidney in yellow. And you can generate those 3D images. So as we'll see, we can then work and visualize it in every single direction. We can also virtually slice it. So you preserve the integrity of the embryos, and you can really study very carefully how the different ducts are organized. So next slide. So what we've published a few years ago was just a paper that was describing, for instance, this classic knowledge of the fact that in male, the Wolffian duct will stay and the Müller duct will degenerate, and that in female, it will be the other way around. The Müller duct will stay, as you can see, and then the Wolffian duct will degenerate. So in the past few years, so next slide, people in my lab, so you can click, yeah, maybe we see their face. Two postdocs in the lab, Chad Moore and Edwin, both great postdocs, and together with a technician, Ali, and I should also mention Anaïs, who has been really helped a lot establishing the new light sheet system in our lab. So all of them together started to use and to apply this technology to different questions. One of them is, can we use it to study the development of human genitalia that was not studied so well before? So, and I'll show you here just one example of what has been done using like electron microscopy, but it's not very precise. So the next slide will show you the type of image we can get. So here, if you play the movie, it's a movie that was obtained using like a virtual reality system, a software named SciGlass. And here, what Ali has done, we stained it with an antibody against the smooth muscle actin. So what you're seeing now are all the arteries in 3D. We're moving the sample in VR. And what Ali has been doing is to use this software to paint all the arteries that are going to the genitalia. So what we're doing this at different stages. We want to build a map of the innervation, of the vascularization, of the developing gonads and genitalia. And this type of tool is very useful to address this type of question. So here we're seeing arteries, but of course the innervation is much more dense. So the next slide will show you how the vasculature look like if you stain now the endothelial cells. So next slide, please. Yeah. So here again you have the penis, and it stains with PLVAP, a marker of the vasculature, and you can see the density. And of course, if you want to extract some information, understand how it evolves between male and female, what we're doing here, so it's to use a software that was developed for understanding organization of blood vessels in the brain. It's named TubeMap. And here, what you do from the raw image, you extract a skeleton. So now you'll be able, from the raw data, to extract the skeleton and to quantify the number of branch points, the density of the innervation. So even if, as you can see, it's very, very complex and dense, you can still try to see what happens between male and females. When do you have the first evidence that there's a difference in the vascularization in the genitalia? So as I said, we also look at the nerves. So next slide, we show you examples of a few samples that were stained for the vascular marker and also the axonal markers. So you can see all the innervation across time. I've been collecting many samples, now between 5.5 weeks all the way to now almost 20 weeks. The next slide, we show you movies again of how it looks and the resolution. So you can play the two movies on the right and on the left. So there's a male and a female. You can see it looks very similar again at that age. So we are around 9 weeks. And you can see in blue the nerves and in magenta the vasculature. So again, for the innervation of genitalia, it was not well described. So how does it start? How does it look before sex is determined? And how does it look after sex is determined? And also what occurs, what is the remodeling of the axon that occurs during development? So the next slide, we show you the type of analysis we can do. So here, if you play the two, you have the raw data, and then we can use again software to try to isolate virtually the different nerves that are innervating the penis or the clitoris. So here you have two examples. So they are like pseudo color, and here we can follow over time the different branches that are innervating the organ and just see what is the density, or do they get remodeled when the genitalia are getting differentiated. So next slide, we show you just an example of a male, where here the same colors are used, and you can see how it looks at 6 weeks, 7.4 and 9.1 week. So of course the analysis is not done, but now we have enough data set to try to understand how the innervation develops and how the different layers and parts of the genitalia are getting innervated. So we're using a lot of we're also trying to realize other cell types. So the next slide, we show you a movie where we will see the nerves again, and the vessels. And in yellow here, what you're seeing, they are the Schwann cell precursors. So Schwann cells are forming myelin around nerves, and during development, they're born close to the spinal cord, and they migrate along the nerves. So here you can see the yellow cells that are associated with the blue, and they will form a very dense network. So we're trying like that to visualize different cell types in the developing genitalia. So we're not only studying the genitalia, but we're also, of course, studying the gonads. So the next slide, please, will show you an example of all the markers we've been trying. So, of course, we've used already a lot of antibodies, and we validated them for 3D to visualize a lot of cell types. So if you click, you will play the movie, and also here what you see is OCT4, which is a marker for the germ cells, SOX9, a marker for the Sertoli cells, and CoopTF2, which is a marker for the support cells. And you can see here that the type of resolution we can get. We can now see very nicely the nuclei, and we are making like this a database for many cell types. So we'd like to be able to quantify every single cell types in the developing gonads at different stages. So the next slide, we show you examples of markers that we've tested and validated. So yeah, so next one. Yeah, so here you have examples of different, if you click once, they will all play. Yeah. So here you can see like many of the markers we tested. And again, we will put online a library of data that we'll all be able to access when we're done. But we have like this, a lot of markers that were validated. So it's nice to be able to visualize the cells, but we like to be able to get more information. So the next slide is showing you that we have also been trying. So when I say we, it's Edwin Hernandez, a postdoc in the lab, who has been trying to develop ways to count the cells in 3D. So here on the top left, you can see the original image. And then he's using like artificial intelligence and neural network to count the cells. So that's why we use a lot of antibodies against transcription factors, because then the nuclei are very well delineated. So through a pipeline that Edwin has been using, and we use also a lot of commercial software, such as Avia, that now belongs to Leica, you can go from the original image, and after deep learning, you can generate this image on the right, where every single nuclei is now isolated. And now he's applying it to a lot of samples with stain. And you can see in the bottom right, he was able to count here in testis, exactly how many OCT4 positive cells you have on the left side, so 27,000, and on the right side, more or less 28,000. So we hope, like this, that we're going to be able to describe really the time course of the development of all the main cell types in the testes and ovaries. So next slide is just to thank all the people in the lab that are involved in this project. So Ali Apiagou, I mentioned before, Anaïs Favre, who is doing also a lot of stainings together with Edwin Hernandez, Chad Moore, as well as Gerard Coulier, an MD, is also helping us with all the embryology. The next slide. Here, I want to acknowledge also, as I said, we work in a large consortium together with groups at the Sanger Institute in Newcastle, and as Anifa at the Karolinska Institute, and also in France, in particular in Rennes and in Lille. So the group Paul Giacobini and the group for Antoine Roland, Frédéric Chalmel, and Cyril Mazodito. And the next slide is just, I'd like to mention what we are organizing a meeting in Paris on May 12th. There's still a few spots you can join. It's free. So it's not virtual. You have to be in Paris, which is not so bad. And so again, you can still submit your, you can still register for the meeting until next week. And email me if you need more information. Thanks a lot for your attention.
Clap, clap, clap. Really well done. I mean, amazing images, Alain. It was very impressive to see all of this together. So I will start with some questions, and please remember to put some questions in the chat. So you mentioned
Ellen, do you want to moderate the chat? It looks like Roser lost connection.
Yeah, we lost her. Okay.
Okay, can you hear me now?
Yeah, exactly.
Oh, there you are. Yes, I can hear you, Roser.
Sorry. Yeah, I was. I don't know if you hear me, but I was thinking. I was asking about the integration of your amazing images with other technologies like single-cell genomics, which is also being generated. How are you thinking that this can be done?
If it can be done, it's complex. It's going to be difficult. We tried for now even to do spatial transcriptomics, and it's hard to combine the. It seems that the clearing and the spatial transcriptomics, they don't go well together. So I'm not sure. We'll see. We keep trying other methods, but it's going to be complex to like do spatial transcriptomics in 3D, for instance. So that's going to be difficult. So it might be hard to combine everything. What might work is, and that's also something you're trying, and for now it's not, also we need to do more, is to try to see whether we can image in 3D, then cut again, and reprocess to do like spatial transcriptomics, which would be useful. If you can extract cells from a cleared sample to do further analysis, it's something we haven't tried. Our work from Ali Erturk, where he showed that from a cleared sample, you can do proteomics. So you can clear your sample, you can stain it, then you can go with a needle and extract 50 cells and do proteomics. And he said in his bioarchive paper that the quality of the sample, the results are equivalent to what we get from a fresh tissue. So maybe it will be more the type of thing we do, map in 3D, and then go somewhere, extract the cells, and maybe do proteomics. Somehow this technology preserves very well the proteins because we fix them. So that would be...
Yeah, that makes a lot of sense. And also what you say about having a spatial transcriptomics as the middle point between these 3D images with 4 markers and the single cell, and in this way trying to integrate everything. I have another question from Anne Harris. She congratulates you for the beautiful talk. And also she asked about, she says that the sperm maturation depends on the epididymis. And she asked, are you also looking at the genitalial ducts?
Yeah, we stain everything. So we have different markers, and we look in the whole system. So the innervation, the vascularization, the lymphatics, the immune system, everything is integrated. So yeah, so of course the ducts would be also included, but I couldn't in 15 minutes present everything. So the goal is to redescribe everything as much as we can and as late as we can. We haven't started yet to stain like adult testes or adult ovaries, but it's something that we will try for sure.
Yeah, that would be amazing. So Sue Hammond is asking, well, she's again amazed by your images. And she asked if you can elaborate on segmentation and quantification. So she says, I am presuming that you are counting germ-soma ratio.
Yeah.
What pan-surface marker do you use in the testis?
So we're counting everything. So now we have markers that we have defined very well markers for like Leydig cells, Sertoli cells, most of the main cell types. We're trying to count everything at different stages. And also we're trying to map them in 3D and to make like a map of the distribution of the different cells. So like heat map or density map. So to have like to try to describe, including also the test codes. So for the surface marker, we did some staining with like EPCAM, and that is also quite nice. And otherwise, we use SOX9 and a bunch of markers we can use. And we're trying to see also all the test codes form in 3D, but it's again for now it's a bit early to give results.
Thank you so much again. Fascinating images and fascinating talk. I think we don't have any more questions, so I will let the other speaker. Thank you and see you later.